My name is Urs Meyer. I'm an associate professor at the Institute of Veterinary Pharmacology and Toxicology at the University of Zurich. And I'm speaking here on behalf of the awardee, Joseph Scarborough, who was a former PhD student in our group. And he's now a postdoctoral researcher at Chapel Hill University in North Carolina. And so we're working in the fields of behavioral neurosciences and pharmacology. And we're mostly interested in animal models of neurodevelopmental disorders. Now, we use these animal models in order to address the questions of how certain environmental adversities can shape brain development and behavior, but we also use these animal models to test new pharmacological treatments against these disorders. Joseph developed and validated a novel oral administration method that can be used for small laboratory rodents such as mice. And it is termed the micropipette guided drug administration procedure, quite a long name, so in short it's called the MDA procedure. And he has shown that in various experimental series that this method can be used as a non-invasive and minimally stressful alternative to the current gold standard, which is at the moment still oral gavage. And as you may know, oral gavage means that it's an involuntary insertion of a feeding needle into the oral cavity, into the stomach, and as such obviously is invasive, can be very stressful to the animals, and can lead potentially to tissue damage. And so novel techniques are urgently needed, especially for chronic and oral pharmacological treatments in small laboratory animals such as mice. And the MDA procedure, we hope at least, is going to be such a procedure, an alternative to oral gavage, because it's non-invasive and minimally stressful. And the main principle is actually quite simple. It is based on the presentation of a palatable vehicle solution, which takes the form at the moment of sweetened condensed milk solution. And because of its palatability, animals without any sort of manipulation, such as food restriction and so forth, voluntarily ingest that solution with minimal training. And so that's the principal idea behind the MDA, that we use a palatable vehicle solution that the animal will voluntarily ingest because of the palatability. So the development of the MDA method has practical impacts or significance, especially for the refinement aspects of the 3 Rs. As such, it will refine existing procedures. Mostly we hope to establish, in some settings at least, the oral gavage, which is much more stressful than the MDA. And yeah, so that's the idea. And I think with further establishment and dissemination of this MDA method, we're pretty convinced that it will be a very good alternative or addition to the oral gavage, and therefore will have practical significance, especially for the refinement aspects of the 3 R principles. Getting new techniques on board is that researchers really stick to what they know best. So to procedures that are around for years, and also procedures that have been validated thoroughly. And I think the challenge really is to boost awareness to researchers that there are new techniques, especially in our domain, which will be novel administration techniques. Maybe the main reason why it is difficult to implement new techniques in our field is, well, actually there's 2 reasons. First, people might not be aware that novel techniques exist. But secondly, as compared to validated procedures, like those procedures that exist for a long time, new ones may not be as validated thoroughly. So I see it very important for the field to advance 3 R in general, that we thoroughly validate new techniques using really sound scientific research in order to sort of convince people that these new technologies are equally valid and reliable as the old ones. Practically, we would like to further validate the MDA method precisely because of the reasons I just said. Namely, I think it's very important that we can convince other researchers that the new techniques are scientifically solid. That's why we need to thoroughly validate them. And as an example, we will, for example, validate the MDA method in terms of pharmacokinetic profiles. We will, for example, compare different vehicle solutions. We will look into species differences, sex differences, and so forth. So certainly there's a lot of open questions that we'd like to address with future research. And on the other hand, we'd also like to disseminate the MDA method more thoroughly. And we will do so in collaboration with various organizations. For example, we're in close collaborations with, for example, animal welfare officers or representatives of the 3R nodes at the University of Zurich. And with the help of these sorts of collaborations, we try to establish the MDA method and disseminate it more thoroughly amongst other researchers. And also in general, I think it's important to try to extend the applicability of the MDA procedure to various research fields. So far we have done it mostly in the domains of behavioral neurosciences and neuropharmacology, but we'd like to see whether the MDA procedure can be used as a valid oral administration techniques in other research domains. Lucky enough, we had recently received a funding from the National Research Program 79, which is in the context of advancing 3R. And so we were lucky enough to get funding to precisely do our homework and validate the MDA method so that we can hopefully answer the remaining questions.